Good morning, everyone. Myself, Madhuri Vaishampayan, Assistant Professor, Department of Biotechnology, Changu, Kana, Thakur Arts, Commerce and Science College, New Panvel, Autonomous. Today, we'll introduce our students with the basic concepts of molecular biology. We'll start with the structure of nucleic acid. Now, whenever we are talking about nucleic acids, this is the language of your DNA and RNA molecules, which are having the very huge information about the genetic information which is transferring from one generation to next generation. So, nucleic acids are nothing but what? They are the polymer. They are the large biomolecules which are essential for the different life forms. They are basically categorized into the two types. One is your DNA molecule and another one is your RNA molecule. Now they are composed of your nucleotides. Now whenever we are talking about the nucleotides, it is the mixture of three important components. Now what are these components? The first important component is your 5-carbon sugar. Second important component is your phosphate group and third important component is your nitrogenous base. We are going to study each and every component one by one. Now whenever we are talking about the sugar which is present in your DNA and RNA molecule, they are basically of two types. One is your ribose sugar and second one is your deoxyribose sugar. Now when the ribose sugar is present, the polymer is known as RNA molecule. When the deoxyribose sugar is present, the polymer is known as DNA molecule. This is the short history where we have started with the nuclein and we have reached up to the structure of the DNA. Now, it is started with 1869. The nuclein, the term was discovered by Friedrich Mischer. In 1880, the Albert Kochel, they have further purified the substances and identified their highly acidic properties. In 1889, the Richard Ertmann, they have created the term nucleic acids. So in 1938, Asst and Bell, they have published the X-ray diffraction pattern of DNA molecule. In 1953, the Watson and Crick, they have determined the structure of your DNA molecule. Now, whenever we are talking about the structure of the DNA molecule, it is basically consist of your nucleotides. Now, whenever the term comes nucleotides, it is made up of nucleoside and your phosphate group. Now again, nucleoside is not a single component. It is a mixture of your nitrogenous base and your pentose sugar. So in cumulatively or commonly, we can say it as the nucleic acid is the mixture of nitrogenous base, pentose sugar and your phosphate group. Nitrogenous bases. So these nitrogenous bases are the nitrogen containing bases. Okay, so chemical properties are basic in nature. So they have the different functions in the nitrogenous base. Now nitrogenous base are classified into two. One is pyrimidine and another one is your purine. Now whenever we are talking about purine, purines are nothing but what? They are your heterocyclic aromatic organic compound. Now they are consist of the two rings and they are water soluble. Purines are having the two different chemical structure. One is adenine and second one is guanine. So here you will be able to see the two structures of adenine and your guanine. As like your purines, in case of pyrimidine, it is a single ring structure and this is your heterocyclic organic compound. It has the nitrogen atom which is present and at first and third position of the ring. This is the second important component of your nucleotide, that is your pentose sugar. Now pentose sugar, this is your monosaccharide. Monosaccharide with 5 carbon atoms. So the total number of carbon atoms which are present in the structures are 5. 1, 2, 3, 4 and 5. Now in case of your pentose sugar, the two types of sugars are present. One is your ribose sugar and second one is your deoxyribose sugar. This this sugar or this pentose sugar will play a very important role in deciding whether this is your DNA or whether this is your RNA. Why I am saying so? Because if the deoxyribose sugar is present, it is involved in the structure of your DNA, and if ribose sugar is present, it is involved in the formation of the structure of your RNA molecule. Now, this is the schematic representation of nucleoside and nucleotide. This is your nitrogenous base, and to this nitrogenous base, your phosphorus groups are attached. Now, if one phosphorus group is attached, then it is known as nucleoside monophosphate. If two phosphorus groups are attached, it is known as nucleoside diphosphate. If three phosphorus groups are attached, it is known as nucleoside triphosphate. Now, the important concept is related to your polynucleotide. Now, poly means what? Mono means your single unit. When many single units which are joined together, it is known as your polymer. So, in case of your DNA, that is your deoxyribonucleic acid, and RNA is your ribonucleic acid. These are the best examples of polynucleotides with the distinct biological functions. These nucleotides are held together with the help of phosphodiester bond. So this phosphodiester bond plays a crucial role in formation of DNA or RNA polymer. Now, whenever we are talking about this phosphodiester bond, it occurs with the 2-hydroxyl group. Now, this 2-hydroxyl group present in the phosphoric acid will react with the hydroxyl group of another molecule, and then they will form the 2 ester bonds. So, these phosphodiester bonds are central to all life on Earth, right? So, the DNA and RNA, these are the phosphodiester bond is the linkage between your 3rd carbon atom of one sugar and 5th carbon atom of another. So this is how the phosphodiester bond is formed. This is the diagrammatic representation of how the phosphodiester bond is formed. This is the comparative study between your DNA molecule and your RNA molecule. Now, DNA is your deoxyribonucleic acid and RNA is your ribonucleic acid. Now, when question comes to the stability as compared to your RNA, DNA molecule is very much stable and it is commonly found in nucleus, mitochondria as well as in case of your plastids with respect to your plant cells. RNA is commonly found in nucleus as well as in cytosol. So, whenever we are talking about the structure of the DNA molecule, it has the long nucleotide chain. The 2 complementary strands are present. Now, in case of your RNA, this is your short nucleotide chain with 1 strand. Now, when question comes to your backbone, in case of your DNA, the backbone is of your deoxyribose sugar, and in case of your RNA, the backbone is of your ribose sugar. Now, in the same way, there is a difference in the nitrogenous bases which are present in your DNA as well as in your RNA molecule. In case of your DNA, we have a combination of thymine, cytosine, adenine and guanine. In case of RNA, your thymine is replaced by uracil. So, rest of the 3 bases, nitrogenous bases will remain same. Only the thymine will replaced by uracil. In case of your complementary base pairing, A always pairs with T and G always pairs with C. Now, G always pairs with C is same in case of your DNA as well as in case of your RNA structure. But whenever we are talking about A and T, it is commonly found in your DNA molecule, and A will complementarily having the pairing between your uracil with respect to RNA molecule. Now, in case of your composition of your DNA molecule, basically DNA is your double helical structure. Now, whenever we are talking about the double helical structure, it consists of 2 different strands. Now, these 2 strands are coiled together to the imaginary axis. Now, when we see about the orientation, then these strands are anti-parallel in nature. Means one strand runs from 5 prime to 5 prime to 3 prime, and another strand will run from 3 prime to 5 prime. The sugar phosphate backbone, these are the sides of the ladders which are formed by the sugar, and the phosphate groups of each nucleotide, they are linked together by the chemical bond. Now, the nitrogenous bases which are present in your DNA molecule, they are your adenine, thymine, guanine and your cytosine. In case of your complementary base pairing, these purines and pyrimidines, they show the complementary base pairing with each other. So, adenine always pairs with thymine with the help of double bond, and guanine always pairs with cytosine with the help of triple bond. So, which bond is present between these nitrogenous bases? The hydrogen bond which is present between your complementary base pairing. In all species, it is composed of the two helical chains which are bound to each other with the help of hydrogen bond. Now, if we consider the pitch of your DNA molecule, it is 34 Armstrong units or 3.4 nanometers, and the pair of chain has the radius of 10 Armstrong or 1.0 nanometer. Now, this is the diagrammatic representation of the DNA structure. These are the two strands. These are the complementary base pairing which is present. This is known as your major groove and this is known as your minor groove of DNA strand. So, this DNA may exist in A form, B form and Z form, but commonly in living organism, B and Z form is functional. This is the structural variation between your A, B and Z form of the DNA molecule. Now, we see the composition of your RNA molecule. As we have seen the DNA molecule, DNA means what? Deoxyribose nucleic acid. Now, this is RNA means what? Where the sugar which is present is your ribose sugar. So, the name will indicate ribonucleic acid. So, this is your polymer of the molecule which is essential in the various biological rules. Say for example, coding, decoding, regulation, expression of gene. Everywhere this RNA molecule will play a very crucial and important role. Each nucleotide of your RNA will contain again ribose sugar with the carbon number of 1 prime through 5 prime, and the bases which are attached to 1 prime, they are your adenine, cytosine, guanine and uracil. Now, there are multiple combinations which are available. So, this adenine and guanine, these are your purines, and cytosine and uracil, these are your pyrimidines. This is the diagrammatic representation of your RNA molecule. This is your single stranded structure. This is your uracil with a single ring structure, adenine and guanine with double ring structure, and again cytosine is having a single ring structure. Complementary base pairing will remain same. A will pair with U and G will pair with C. There are 4 different types of RNA molecules which are available, mRNA, tRNA, rRNA and snRNA. mRNA is your messenger RNA. tRNA is your transfer RNA. rRNA is the ribosomal RNA and snRNA is your small nucleolar RNA. Now, mRNA will encodes the amino acid sequence of your polypeptide. This is the major role played by your mRNA molecule, and transfer RNA will bring the amino acids from to ribosomes during the process of your translation. Ribosomal RNA is having the ribosomal protein which are make up of ribosomes and organelles that translate the mRNA molecule. snRNA is your small nucleolar RNA with proteins. They form the complex which are mainly involved in the RNA processing in case of your eukaryote, and this is not commonly found in your prokaryotic cell. This is the diagrammatic representation for your mRNA molecule. This is your transfer RNA or tRNA, and this is your ribosomal RNA or your rRNA. These are some references. Thank you.